We're going to continue talking about the basic building blocks that make up biological membranes. And lipids don't get a lot of attention, but you'll see that they can form very diverse classes of structures with very different important properties. In this lecture, we are going to look at the structure as well as how you can name lipids. There's quite a bit of nomenclature that you need to understand about lipids, and historically scientists may have many different ways of naming the same molecule, but we will go over the general ways we can describe a lipid, and I will highlight the main structures that you need to know. We'll review the role that lipids play in a cell and see how their chemical properties play a role in their function. We'll also look at a few examples of their importance in health and disease. These are the supplemental readings for this lecture. We're going to talk about some of the ways of classifying lipids, and you'll see that this class of molecules is very different than proteins. Where proteins have amino acids linked in a chain, lipids are a little bit more nuanced than that. They can have very different structures and are defined by their similar properties. This allows them to provide structural support for the cell, and they can also store carbons for energy in fat tissue. And remember, they do play a role in signal transduction as well. We call something a lipid based on its properties rather than what it looks like. So these molecules tend to be less soluble in water, but highly soluble in nonpolar solvents like octanol. I'll show you some structures in the next slide, but you'll see that they are quite diverse, and some of them might even be completely hydrophobic, while others are amphipathic, meaning they have both polar and nonpolar hydrophobic groups. Most biological lipids will fall in the second class, since it's really important for membrane formation. These are a few examples that show you just how diverse these structures can be. You may notice some common features, so there's lots of hydrocarbon tails, and you can also pick out a few polar functional groups, so some hydroxyls, phosphates, maybe amino groups, and some of them, like these three structures, are completely hydrophobic. Typically, we do draw them as lines, but remember that the space-filling model shows all of the atoms, and there is 3D geometry that will be important. So really the take-home message is that this class of molecules is not defined based on their structure, but more their properties, and specifically their insolubility in water. We've already seen how lipids self-assemble to form a bilayer, and there really isn't anything else that puts them together. This is a spontaneous process where they aggregate to avoid water due to the hydrophobic effect. We see that hydrogen bonding occurs between the polar groups in water, and you might even see some van der Waals packing happening between the tails. In addition to the bilayer, keep in mind that other structures can form, such as a monolayer, which you might observe if you try mixing oil and water in your kitchen, or other things like a micelle where all the tails come together in the middle of a droplet, or an inside-out micelle that has the tails pointing out with a small amount of water on the inside. When a bilayer comes together to form a vesicle or a cell, if it was a vesicle, we sometimes refer to this as a liposome of pure lipids, and it really depends on what types of structures and the properties your lipids have that determines what structure forms. For instance, if it has one tail or two, that will allow these different types of macromolecules to form. Biochemists love to classify things, and these are the major classes of lipids. Fatty acids are the simplest lipids, and they can come together to form triacylglycerides, which is how we store carbons as fats. Many other types of lipids either contain or are derived from fatty acids. The phospholipids will contain a phosphate group, hence their name, and along with sphingolipids that have a sphingosine backbone are part of the structural lipids. Glycolipids, which we've already seen before, have monosaccharides attached to these two backbones, and they're also part of the structural lipids along with sterols like cholesterol. Eicosanoids are also derivative of fatty acid chains, and these, along with cholesterol and other steroids, can be involved in signaling. Now, you may have heard of steroid hormones like cortisol and testosterone, and these really are lipid molecules. So all of your other courses like physiology are really based on biochemistry. That is the chemistry of these biomolecules, allowing them to carry out their functions. Let's start with the basic unit, and that's the fatty acids. The fatty part includes a hydrocarbon chain that's usually anywhere from 2 to 22 carbons long, but chains can be as long as 36 carbons. They frequently contain an even number of carbons, and that's based on how they're made in a cell. Now, these chains can either be saturated with hydrogens, as shown, or contain double bonds and be unsaturated, and this will have important implications for their properties. We do number the carbons down from the carboxylic acid group, which would get priority, but you may sometimes hear about omega fatty acids, and these are identified based on where their double bonds are relative to the terminal methyl group. Free fatty acids also have a carboxylic acid group, and similar to what we've seen before, this group has a pKa around 4.8. So what do you think would be the protonation state of this free fatty acid in a cell? If you said deprotonated, you're right. And so there really are common themes that keep coming up in biochemistry, and this is why we reviewed all the functional groups and pH at the beginning of the course, because they do play important roles. So this portion, the carboxylic acid group, is the hydrophilic part, but it can also be joined to other molecules, and we'll see that ester bonds include that oxygen linking a free fatty acid to other molecules. There's also some amide bonds that'll form, and we'll see an example of that later on. When it comes to the nomenclature for fatty acids, you'll sometimes see them written out as two numbers separated by a colon. The first number is the number of carbons in the chain, and this includes the carboxylic acid and the methyl group at the end. And then the second number is how many double bonds are present. So 18,0 would be a saturated chain containing 18 carbons. The systematic name would be N, where N means normal or no double bonds, octa meaning 8, deca meaning 10, noic acid for the carboxylic acid group. When double bonds are present, a monounsaturated fatty acid would have one double bond, where polyunsaturated fatty acids will have multiple double bonds. On the right are images showing the differences in structures that occur when you introduce a double bond. So here's the linear chain that we would normally draw, but do remember that these atoms do take up space, as in this space-filling model. When you introduce a double bond, biological fatty acids usually contain the double bond in the cis form, and this will create a kink that can be seen on the right. If we were to write out the systematic name, it would be cis delta, where delta describes the location of the double bond relative to the carboxylic acid. Octa, decene for that double bond, O8, where 8 could demonstrate that it is the basic deprotonated form. 18,1 would be the short form, and you could also call it an omega-9 since the double bond is on the 9th carbon from the methyl end. This table just shows you the different types of chains that can exist and their names. You don't need to memorize them. We would give you the number of carbons and double bonds, so the shortened version such as 18,1 is what you'd be given. Okay, but do keep in mind that we do have different ways of describing them and that they'll all have different properties. Now, the degree of saturation is really important for membrane structure and its fluidity. So what kind of forces do you think exist between these lipid molecules? We learned previously in this course about ionic interactions, such as salt bridges and hydrogen bonding, and these forces will hold these head groups together. Van der Waals forces can exist between the tails. So the extent of packing between these tails really depends on the degree of saturation. On the left is a group of fully saturated fatty acid chains, so there's no double bonds, and they can pack together in a very orderly manner. You can imagine that there isn't much fluidity or movement within this structure. There's lots of van der Waals packing occurring. We can measure the amount of energy needed to melt or separate these strands, and this would actually result in a higher melting temperature. On the right is a mixture of saturated and unsaturated chains that would allow for membrane fluidity. It would take less thermal energy or a lower melting temperature to disrupt the packing between these lipid-lipid interactions. And if you were to introduce a membrane protein, you could imagine that it would have the flexibility to undergo any conformational changes needed to carry out its function. So when we talk about the fluid mosaic model, it's the presence of these cis double bonds within the fatty acid chains that can help maintain fluidity. And if you want to store carbons in the chain for energy, such as in fat cells, you would want these saturated chains as shown on the left so that you can pack as many in. And this is what we do see for those triacylglyceride molecules in fat tissue. One way we can characterize lipids in biological membranes is by measuring the phase transition. So looking at how much energy is needed to go from that gel-like solid phase to a liquid crystalline phase, kind of like if you were melting the membrane or a sample of lipids. This melting temperature is an index of the fluidity, so how much energy is needed to separate the strands, where a lower TM would indicate a more fluid structure. A higher TM means you need more energy to disrupt the forces that are holding these tails together. You can do these melting temperatures on pure samples of liquids, so say all C18-0 chains, and you'll get a very sharp phase transition. While biological membranes will have much broader peaks due to the heterogeneous nature of the membranes. So I already mentioned this before, but in order to store carbons for energy, they are stored in fatty acid chains, and these would be linked by ester bonds to a glycerol molecule. This is what gives them the name tri, meaning three. So you'll have three acyl for the chains attached to glycerol, a glycerol backbone. And you may also hear the word triglycerides used interchangeably. So this is how we generally see fat stored in adipose tissue, and it is a very hydrophobic dehydrated molecule, so you can pack quite a bit in, allowing for the storage of carbons to be quite unlimited. A mixed triglyceride, as shown in the top right, can have double bonds in the chains, but we usually do see this saturated structure since it does allow for the tight packing and more efficient storage of fat molecules. When you look at the naturally occurring fats that exist in nature or even in your kitchen, you'll see that most are mixtures of different types of fatty acid chains. The length and level of saturation affects their melting temperature. So on the right is just a graph showing the different compositions of three common fats that you may have heard of. So since olive oil has mostly unsaturated C16 and C18 chains, shown in green, it takes less energy to melt or disrupt the non-covalent interactions between the chain, and this is why it's usually a liquid at room temperature. Beef fat, on the other hand, has longer saturated chains, and so these C16 and C18 saturated chains, so it is a solid at room temperature, and you would need more energy to melt the chains. Butter is kind of in the middle, where it has a mixture of all these different fatty acid chains and can be a solid or liquid depending on the temperature. So these three are all mixtures of triacylglycerols that differ in their fatty acid composition. And it's the chemical properties that result in their different melting points, and this also has implications for our health. I've also mentioned the omega fatty acids, where omega indicates the location of the double bond relative to the methyl end. Shown here are two of the more common omega fatty acids, and they are polyunsaturated fatty acids. 22,6 would have 22 carbons and 6 double bonds. Now it would be an omega-3 since the first double bond is on the third carbon relative to the methyl end. 18,2 is an omega-6 fatty acid since its first of the two double bonds will be on carbon number 6. You hear about these all the time since they are considered essential in that your body can't synthesize or make these fatty acids within a cell. We do need to get them from our diet. Now, these types of fatty acids do play a role in the signaling pathways as well as in inflammation. And it's not that one is more important than the other, so omega-3s versus omega-6. Researchers have shown that you really do need a balance of all the omega fatty acids. So you can either take supplements or get them from natural sources. And just keep in mind that when it comes to all of these biomolecules, you really do need a balance of them in order for your cells to carry out their particular function. On the flip side, the food industry has been extensively studying these fats since working with them can be a little bit tricky based on their properties. So what chemists have discovered is that you can change their properties in order to make them a little easier to work with or affect their shelf life. We can remove the double bonds in this partial hydrogenation process, which will create fats that have trans double bonds, such as seen in oleic acid. And this can make them great for baking, but we now have a trans double bond, and this actually results in a higher melting temperature. And that's what allows them to pack together and can clog your arteries. So there's been a big movement to remove these trans fatty acids from your food, and this is why you can see their difference in melting temperatures, so going from 13 to 43 degrees. And if you think about what body temperature is at 37 degrees, you can now understand what's going on at the molecular level, allowing these molecules to pack tightly together. The lipids that we see in biological membranes can have very diverse structures, but remember they are amphipathic molecules that will have these hydrophobic fatty acid tails attached to polar hydrophilic head groups. The two main types that we see are glycerol phospholipids, as shown on the right, as well as sphingolipids, and we'll look at these structures in a bit. And there is quite a bit of variability depending on what chains are present, so either in their length or saturation, as well as what functional groups are found in the polar head group. Cholesterol is the most common steroid found in membranes, and it does have this fixed structure where 4 fused rings are linked to a hydrocarbon tail, and it's really only the single hydroxyl down here that gives it any hydrophilic properties whatsoever. The glycerol phospholipids always contain 2 fatty acid tails attached to the C1 and C2 positions on a glycerol backbone. If we examine the commonly occurring glycerol phospholipids in membranes, you'll usually see an unsaturated chain at C2, so this tail here will have a double bond present. The functional group at C3 will include a phosphate linked to different molecules, and these are shown in the table. If there isn't another molecule present, so just a hydroxyl on the phosphate, we would call this phosphatidic acid or phosphatidate for the conjugate base. Phosphatidylethanolamine, or PE for short, has ethanol and then an amine group attached, and this positive charge neutralizes the negative charge that we would see on the phosphate, resulting in a polar but net neutral glycerol phospholipid. Phosphatidylcholine, or PC for short, will have a choline group bound and also be neutral in charge, while the amino acid serine can also be attached to form phosphatidylserine, which would have a net negative charge due to serine's positive and negative charge. If you remember from the last lecture, we talked about how flipases can move PE and PS to the cytosolic leaflet, resulting in asymmetry across the membrane. Now, PE is neutral, but overall PS is negatively charged, so this actually gives the inside of the membrane a net negative charge. You can also have glycerol bound or 2 glycerol phospholipids linked to form cardiolipan or diphosphatidylglycerol, and the addition of inositol, a sugar derivative, can also be found in membranes, resulting in this molecule inositol 4,5-bisphosphate. Now this molecule is involved in signaling pathways, and it also contributes to a net negative charge on the inside of the cell. If you look, you would count actually only 3 negatively charged phosphates, but if you remember the pKa values for phosphates, there is one near physiological pH, and that's why it's listed as a net negative charge of -4. So these are the common glycerol phospholipids, so do make sure you can recognize them and know their properties based on what the head group is that's present. There's always going to be that negatively charged phosphate on the backbone and with glycerol bound to 2 tails, but depending on what head group is present, you can have different properties arising, such as charge. The sphingolipids are based on a sphingosine backbone instead of glycerol, giving them unique properties. Sphingosine is a long-chain alcohol with an amino group at C2, so it actually has its own tail built in. Ceramides have a long-chain fatty acid group that's going to be attached to that amino group of sphingosine. Now this is the exception to the rule, where we have a fatty acid tail that's attached by an amide bond at C2, not an ester, which is what we saw for the glycerol phospholipids. You can then add different head groups, so sphingomyelin would have a phosphate and a choline, shown in blue, and that would be attached to that C1 hydroxyl group. Or you can also see sugar molecules bound, such as glucosyl cerebroside, which has a glucose sugar attached to ceramide, and these can be important for diseases. Gaucher disease arises due to a mutation in a lysosomal enzyme. And remember, the lysosome is important for the turnover of proteins and lipids in the cell. So if there's any sort of disruption to either the formation or the rate of breakdown, this can lead to disease. The structure of the enzyme is seen on the right, and a single point mutation can have a huge effect on human health. When the valine at position 349 is mutated to a leucine, so not a major change in properties, but it is enough to disrupt its activity. This lipid can't be turned over and it starts to accumulate, leading to the buildup in lysosomes. We can see that the various properties of the active site residues, so some of them are charged residues that may help with hydrolysis, while there are other hydrophobic residues that are responsible for binding to the lipid. This valine is found in that binding pocket, so the inclusion of a slightly larger amino acid can affect binding of the substrate. So when you inherit a defect in this enzyme, there are very profound effects, particularly in neurons, which lead to cognitive decline, or hepatomegaly, and this can be quite fatal in children. We see that it's a very subtle change in the enzyme, but it's enough to affect substrate binding, leading to the accumulation of glucosyl cerebroside. What's great is that for some conditions there are enzyme replacement therapies, but it can be quite tricky as you do need to get that wild-type functioning enzyme into the cells that need them. And this enzyme specifically can't cross the blood-brain barrier. So the cost can be quite high, and there is that complication of targeting affected cells to alleviate symptoms. I just want to come back to the importance of cholesterol. It can be metabolized to produce other hormones as well as bile salts that help with the absorption of dietary lipids. And its structure, which you can see on the right, can insert into the membrane and interact with different lipids. And remember, we do see its enrichment in those microdomains called lipid rafts. So the image just shows you how it inserts into the two bilayers. It doesn't span the entire bilayer. It's either in one side or the other. And it's the hydroxyl group that will interact with the polar head groups. Now, the way that cholesterol modulates fluidity is a little counterintuitive. If you put it in a membrane that's quite fluid, this ring system actually slows down the fluidity because it does have a very rigid structure. On the other hand, if you start to cool down a membrane, the inclusion of cholesterol can actually help with fluidity since the fatty acid tails can't closely pack around those rings. So it has two jobs on either end of the spectrum. So it prevents membranes from being too fluid, but it can also prevent them from freezing. We can actually see that the composition of membranes can change at different temperatures. So in some organisms at lower temperature, you'll see the inclusion of cholesterol as well as shorter unsaturated chains to prevent their membranes from freezing and maintaining fluidity. So in addition to modulating the composition of membranes at different temperatures, the types of lipids can also differ depending on their intracellular location. So even though lipids self-assemble to form membranes, there are enzymes that are responsible for synthesizing and creating the lipids for a membrane, and these can help direct which lipids are found in the different membranes. So the image is just showing you on the right the different membranes found within a rat liver cell, and you can see that there's probably about 35% cholesterol at the plasma membrane, whereas in mitochondrial membranes we barely have any. You don't have to memorize these numbers, but just appreciate that there can be different compositions depending on which organelle or membrane you're looking at. The key take-home messages are that the lipid composition of biological membranes is quite variable, with many different lipids contributing to the structure and function of a membrane. We can see that it's the presence of double bonds and the length of the acyl chains, as well as the incorporation of cholesterol that can modulate membrane fluidity. And the TM, or melting temperature, can be used to characterize this fluidity. Most cellular membranes will also have a difference in charge on either side of the bilayer, and this is due to the difference in polar head groups that are found on those lipids. So it all comes down to the enzymes that can regulate the synthesis, movement, and degradation of lipids that is important for maintaining human health. Do make sure you review the learning objectives before attempting the brain break questions.